Okay, hi everyone. Thank you for coming to my talk. Today I'm going to talk about aliens. Well, now that I have your attention, I can show you my actual cover slide. So today I'm going to talk about the effects of biogeography and domestication on a facultative ant-plant mutualism in an ancient alien species. And in agricultural terms, that just means a species that was brought to a new range because it was cultivated there. So before I get into the talk, I want to introduce the mutualism that I've been looking at. So this is the ant extrafloral nectary mutualism. And in this mutualism, the plants produce these knob-like structures called extrafloral nectaries, and the ants visit to drink the nectar. And herbivores often don't like interacting with ants, so the ants will ward off the herbivores from the plant. So this is a resource protection mutualism. An ant gets a resource and the plant gets protection. It's facultative and it's generalized. So most of you at this conference will probably have heard at least one talk about the latitudinal diversity gradient, which just says that as you move from the poles to the tropics, there's an increase in biodiversity. And from this, we get the biotic interactions hypothesis, which says that as you move from the poles to the tropics, biotic interactions, which is interactions with other living organisms, become more and more important as forces of selection, as you see here. So my goal was to look at the anti-FN mutualism across a large latitudinal gradient and see if these interactions match up with the predictions of the biotic interactions hypothesis. So my study system for this project was the castor bean plant, or Ricinus communis. This is a common species in India, and I chose this species because it produces different kinds of extrafloral nectaries in different parts. As you can see here, this is under the leaf, on the stem, and this is the GBIF distribution map of this species. So as you can see, it occurs in most tropical regions and quite a few temperate ones. And it also spans these large latitudinal ranges, making it a good study system to study latitudinal variation in mutualism. This species is also interesting because it's a domesticated species and has this long-standing relationship with humans. In fact, from genomics, we know that castor was introduced to India around 2,000 years ago, which is an order of magnitude greater than what we usually think about with other introduced species. It's also widely cultivated in India, and India is the largest producer of castor oil. So this species was carried from its native range in Eastern Africa through the Mediterranean to India and then to China and so on. So since it's had quite a few generations to be in this new introduced environment, it's possible that it's adapted to the local fauna, which could be herbivores, mutualists, and so on. So in this chapter, I wanted to ask how the EFN ant interaction varies across latitude. And specifically, I wanted to understand if partner diversity is higher towards southern Indian sites, which is closer to the tropics. I also wanted to measure if interaction intensity, which is quantified by ant abundance, is higher towards the south. On the plant side, I wanted to understand if the mutualistic traits of castor plants also vary across a latitudinal gradient, and if there's higher investment in mutualism towards the southern Indian sites. How did I do this? To do this, I sampled 11 sites across the Indian subcontinent, and this spanned around a 20-degree range in latitude. I also sampled an island site, which is also part of India, and another unique thing about this data set is that I didn't sample these sites sequentially. I actually randomized the order in which I sampled the sites so that there isn't like confounding variation in things like monsoons, which also happen over the scale of a few months, in which I also did my sampling. So I measured mutualistic traits and interactions. I looked at EFN counts and EFN areas, and also ant abundances, ant diversity, and herbivory, because you can't study protection if you don't study herbivory. Okay, so before I get into my results, I want to say that these plots are going to be pretty straightforward. They're all mostly regressions, but there's going to be a lot of them. So if you lose the thread at any point, just stay with me, because I'll summarize all of it with a nice plot at the end. Okay, so let's first look at mutualism traits. So this is EFN counts for two kinds of EFNs. These are leaf-based EFNs, and these are the stem EFNs. So as you see here, there isn't any significant trend in variation across latitude. However, when I looked at EFN areas, I found a significant decrease in EFN areas from low to high latitudes, which means more southern sites had larger EFNs than northern ones. Herbivory also increased from low to high latitude sites, with the northern sites showing high herbivory compared to the southern sites. Finally, ant abundance also decreased with increasing latitude, so the southern sites had more ant visits compared to the more northern sites. Ant species richness and diversity also showed a downward trend with latitude, but it wasn't statistically significant. Still in line with our predictions, though. So that was all for my wild sampling. So now, to further unpack this variation that we saw, I wanted to see if there was genetic or maternal variation by planting all these varieties in a common environment to remove the effect of the environment. So I wanted to also consider how the mutualism varies between agricultural and wild varieties. And the expectation is that since agricultural varieties grow in these really managed environments, often in proximity to pesticides, they might have sort of weakened the relationship with ants because they don't really need that protection anymore. So in this study, I set up a common garden in the southern Indian state of Bangalore, mostly because I had contacts there, but also because in my previous field study, I found that Bangalore also had one of the highest ant diversities, visiting castor plants. So in addition to the 11 sites that I showed you in the map before, I had a few more sites from which I collected seeds, and also 14 agricultural varieties in this common garden. So more results. I found that, interestingly, the number of EFNs of both kinds increased with increasing latitude in the common garden. Herbivory decreased from south to north. Northern varieties showed lower rates of herbivory than southern varieties. And ant abundance also followed that trend, where southern varieties were visited by less ants compared to northern varieties. Comparing agricultural and domesticated types, I found that the number of EFNs, interestingly, was higher in the agricultural types, which is the orange bar shown here, and this applied to both kinds of EFNs. However, wild varieties, shown in blue here, experienced less herbivory and experienced higher ant visits. So to quickly summarize all the things that I just told you, in the wild sampling study, I found that there was no significant variation in EFN counts, but EFN areas got smaller with increasing latitude. Herbivory increased with increasing latitude, and ant visits decreased with increasing latitude. Now, I'm going to show you the common garden results in both these columns. So compare this column with the column that I just showed you. So in wild-derived varieties in the common garden, I found that the number of EFNs were higher in varieties derived from northern latitudes or higher latitudes. Herbivory was lower in varieties derived from higher latitudes. And ant visits were higher in varieties derived from higher latitudes. Finally, to summarize the wild domestic comparisons, I want you to compare the arrows that I showed you with what we saw in the wild varieties, so the box plots. So domesticated varieties had more EFNs than wild varieties. They experienced higher herbivory than wild varieties, and they experienced lower ant visits than wild varieties. So what does all of this tell us? In the wild plants, we did see some evidence for higher effect size of mutualism at lower latitudes. And we saw support for the biotic interactions hypothesis for mutualism, but not for herbivory. And this could be because even though there was a high abundance of herbivores, it could have been counteracted by strong mutualism. And this might be why the observed values were lower. Comparing wild and domesticated varieties, there was evidence for stronger mutualism in wild varieties, although we saw higher EFNs in domesticated varieties. And we think that the abundance of EFNs in domesticated varieties may be a vestige of past mutualism, where they once produced nectar but didn't have to and just evolved to stop doing it. Finally, why did the trends of mutualism and herbivory reverse in the common garden? We think that this is because the anti-FN mutualism is activated by environmental factors such as biotic interactions, and this may be why they upregulated EFN production. In terms of herbivory, as we saw in the wild samples, the northern varieties actually had high herbivory, and this may have caused them to evolve investment in chemical defenses, which made them unpalatable to the herbivores in the common garden site. Finally, the sites used in the common garden were a little different from the wild sample sites because we included a few additional sites from the north, and we hadn't characterized the mutualistic assemblage for these. So it might sort of be an artifact of that. I want to end by thanking all the amazing people who made this project possible, tagged along with me from site to site, week after week to collect data, helped me set up a common garden in India, which I don't think has been done before, and helped me water the plants when I had to go away, and of course, my amazing lab mates and supervisor. And I'm happy to take questions. Rowan?
That was a great talk. So I'm wondering if you know anything about tritrophic interactions in this system. So in some cases, when herbivores feed on plants, volatiles attract parasitoid wasps that then parasitize the herbivores. Do you think there's a role for that in maybe modulating herbivory?
There definitely could be. We didn't look at parasitoid wasps in this study, but there's sort of a peripheral study that an undergrad did using these plants, and like he tried inducing responses by damaging it to different extents, and he found that it didn't matter. And we think this might be because this was a greenhouse experiment and the volatiles just spread evenly everywhere and induced everyone. So it's definitely possible, but I don't know if it happened in this study. Yeah?
Really great talk, thank you. I don't know if I'm misremembering, but is it true? I know some species that produce EFNs, they stop producing more of them when they're under attack.
Mhm.
Is this true in this system?
It has been shown by other people that herbivory does upregulate both the number and the quantity of nectar. Is that what you were asking?
Yeah. So is there also, do you have any thoughts about why that there might be a change in like the nature of the EFNs in the domesticated species? Like they seem to be not working as effectively. Do you have any ideas or hypotheses about that?
I just think that, so typically these varieties are bred for high yield, which means high seed production. And maybe initially the really mutualistic ones also produced a lot of seeds. But as they were bred over several generations, the seed production remained because that's what farmers want. But maybe the mutualism was a waste of resources, so they just evolved out of it. But I haven't tested it.
Great, thank you.
Lini? Yeah, so agricultural fields don't seem to have a lot of ants, but there's also some interesting trait stuff going on which I haven't shown here. So agricultural varieties tend to have a waxy coating, which ants seem to hate. So that might also be why they don't associate with ants as much. But they do have a lot of herbivores. Any other? Okay, thank you.
Right, hi everyone. I'm Phoebe Cunningham, and I'm a PhD student in Lee Henry's lab in Queen Mary's in London. And today I'm going to talk about nutrient provision and host regulation in 3 distinct lineages of ants. So I'm just going to start with a bit of background on symbiosis. You've probably heard a lot about that already at this conference, but basically symbioses have evolved several times across the insects, and so here are some key examples. So in aphids, you have symbionts which provision nutrients that aren't available in their diet, which is a very low-protein plant sap diet. And you have tortoise leaf beetles, which feed only on leaf matter, so they need symbionts which provide pectinases to break down that leaf matter. And then you have blood-feeding insects, and blood is notoriously low in certain vitamins, like B vitamins. And so lots of blood-feeding insects have also evolved symbionts. So these are all examples of obligate intracellular symbionts. So these are inside the cells of the host. And we see relationships like that have evolved across several times independently across the ants. So in the very different lineages, so Camponotus, Plagiaulepis, Formica, and Cardiocondyla. And so what's really interesting is that even though these ants have independently acquired these symbioses, they're all housed in the same way. So they're all found in specialized cells, known as bacteriocytes, which are around the midgut. So in panel A, you can see in red the bacteria, and that's really packed tightly around the midgut. And then it's also found in the ovaries and the eggs, which are shown in panel B and C. So this is in a Formica fusca ant. And so what's also really interesting about these ants is that even though they've independently acquired these bacteria, they've all acquired them from the same clade of bacteria. So they're all Sedalis-like bacteria. And so through the process of symbioses, of forming the symbiosis, these bacteria lose lots of genes. So they reduce in genome size, which is shown. So basically the yellow colors in these panels show the obligate symbionts, and the blue colors show free-living or non-obligate. And you can see that all the yellow ones have a very reduced genome size, reduced GC content, and reduced coding regions. So all of these are really strong characteristics that these are obligate symbionts that are formed independently in these ants. So even though they've all formed these independently and they've all lost different genes, what all of the symbionts have retained across the ants is the ability to produce tyrosine. So tyrosine is formed through this quite complex process known as the shikimate pathway, so lots of bacteria can do this. And in this system, all of the symbionts can produce the precursor to tyrosine, and then the hosts can complete that step, either via phenylalanine or straight through to tyrosine. So why do we care about tyrosine? Tyrosine is really important for insects because it's used in the melanization and sclerotization of the cuticle, so the hardening, the thickening. And this study basically showed that when they removed the symbionts from beetles, the wing elytra couldn't form properly, they were pale, and they were deformed. So this is kind of like the lead hypothesis we have for what these symbionts are doing in the ants as well. So that leads me on to my big questions for this project, which is what is the role of endosymbionts in ants and how are they regulated by the host? And then, most interestingly, are these mechanisms conserved across the distinct lineages? But I'm actually not going to talk about that today, so I've kind of oversold the talk. But I'm mostly going to talk about this one species that I'm working with in the lab, which is Cardiocondyla obscurior. So they have a symbiont known as Westerbahardia. It's the oldest of all the ant lineages symbionts, and it's really small. It's only 600 kb. It's 75 million years old as a relationship, and it can do very few things. So it can only really make tyrosine, folate, alanine, and glycine. And so this is a really good system in the lab because the ants are really happy producing queens and thriving in lab conditions. So to really try and get at this question, I've so far used 2 main processes, so looking at the symbiont titer or density, and then also the gene expression. So for the symbiont titer, these are the different life stages of this species. So they have 3 larval instars, a prepupation step where they get rid of their gut, and then 2 pupation steps, and then you have the newly emerged workers and the adult workers. So for the analysis of gene expression, what's really cool about the system is that we can actually treat them with heat and remove the symbiont through that treatment. So we heat them at 31 degrees for 4 to 6 weeks, and you can see, so the symbiont titer or density is on the y-axis, time along the x-axis, and after 2 weeks it's really depleted, and by 4 weeks we can't see any of the symbiont anymore. So that means that we can look at symbiotic and aposymbiotic individuals and compare their gene expression. So I did that across different life stages and casts, so pooling the larval stages, pooling the pupal stages, using newly emerged workers, and then also the queens. So, and then I analysed that using DSeq2 and LIMA. So what we did was actually extract RNA from both the host and the symbiont, so we could try and see gene expression happening in both of the organisms. So the results so far, so going back to those questions, the role of the symbiont, how host is regulating it. So the first thing that I looked at was the symbiont titer. And so what you can see is there's actually quite a dynamic relationship between titer and life stages. So it's highest in the sort of larval and pupal stages and newly emerged workers. And you see this quite dramatic drop at the prepupation step. So that's when the meconium is voided, so when it gets rid of its gut. And so we think either it loses some of the bacteria or damages the bacteriocytes in that process, so it loses a lot of it. And then there's sort of like a rapid recovery that occurs throughout pupation and then into the newly emerged workers. So that suggests that actually the pupation step and the newly emerged worker stage may be a very important for that symbiotic interaction. So the first thing I looked at was the symbiont transcriptome. So this was comparing whether there were differences in gene expression across the life stages of the host in the symbiont genes. And actually what we found was nothing. So basically we found there was no significant difference in gene expression in the symbiont across the host life stages. So at first that was surprising, but actually when you think about the size of this organism, it's only got 600 kb, it can only do a very small number of things, so it's likely that it's just a little machine and keeps doing the same thing all the time, and maybe any sort of regulation in its activity comes from the host. So moving on then into looking at the host transcriptome to really try and get at what the symbiont is doing. So first of all, I just compared the gene expression between symbiotic and aposymbiotic life stages. And again, we found a pattern that was consistent with the symbiotic titer, showing that the most differentially expressed genes were at that pupal stage. So at that stage when we know the cuticle's forming and melanizing, it looks like the symbiotic pupae are doing something quite different to the aposymbiotic pupae. So moving on then into looking at what genes might tell us a bit about what the symbiont is providing the host. So going back to that process that the symbionts can do, this shikimate pathway for producing tyrosine. So as I said before, all the symbionts can produce a precursor to tyrosine, but they need a host gene in order to finish that reaction and produce tyrosine. So that gene is the aromatic amino acid aminotransferase, and actually what we found was that this was significantly more highly expressed in symbiotic larvae and symbiotic pupae, and then not significantly differentially expressed in the workers and the queens. So this is consistent with the idea that the symbiont is providing something that's important during the development of that cuticle in those earlier life stages, and when we see that the symbiont titer is higher. And so zooming in onto that pupal stage that we so far are seeing seems to be important for that dynamic of symbiosis, we also found that there were amino acid transporters that were enriched and more highly expressed in symbiotic pupae as well. So it looks like there's a lot of amino acids being transported and also specifically tyrosine being produced by the host. What we also found, which was interesting, was genes associated with the metabolism and utilization of folate. So folate compounds, things to prevent the breakdown of folate, these were all upregulated in symbiotic pupae as well. And so we know that folate is really important for insect development and can also be produced through the shikimate pathway. So the symbiont can use the same machinery to produce this. And in some other systems, such as the tsetse fly, this is what the symbiont is providing the host. So moving on now to how the host is regulating that relationship, looking at the transcriptome. So what we found was that at that pupal stage where we saw a lot of genes differentially expressed, we actually see 5 genes which are associated with sugar metabolism. So all of these genes are able to catalyze the breakdown of sucrose, fructose, or lactose into these simpler sugars, beta-D-galactose and beta-D-glucose. And both of these can be directly taken up by the symbiont and fed into the shikimate pathway. So it looks like the host is actively breaking down sugars it gets from the environment into something that can be readily used by the symbiont to produce the thing that it needs. So that was really interesting. And then what we also found, which was interesting, was that there was an increase in the synthesis of trehalose. So trehalose is an insect-specific sugar that the symbiont actually can't directly use itself. But we saw definitely a high significance of a synthesis of this sugar and also transport of this sugar around the host. And actually, when we looked at the literature, they've shown, so this was a really great paper by Moriyama and Fukatsu, and they showed that when they dissected the symbiotic organ and grew it in different sugar mediums, that actually trehalose was the best for the growth of the symbiotic organ and production of free amino acids, even though that symbiont also couldn't directly use trehalose. So it's possible that both these systems have a mechanism for utilizing that trehalose for the symbiont. So that would be really interesting to show. And then finally, what we also saw was higher expression in the symbiotic pupae of genes associated with lysosomes and phagocytes. So again, if you think about the pupal stage when the body plan is forming and those bacteriocytes are probably forming, it's likely that you're going to get some cell digestion and reorganization of those bacterial cells, and perhaps that's what these genes are associated with. So that's the work I've done so far. This is the work I'm working on now and doing in the future. So obviously, to kind of validate that those genes are actually involved in the symbiosis, we're going to be doing fluorescent in situ hybridization, so looking at the localization of that gene expression. Is it in the bacteriocytes? And then what I'm also doing at the moment is these diet manipulations. So because we can have symbiotic and aposymbiotic colonies, it means that we can give them restricted protein diets and see how that affects different metrics like brood production or melanization of the cuticle or the metabolomic profile, that kind of thing. So just to summarize, so the findings so far tend to show that pupation seems to stand out as a key stage for symbiosis in both the titer and the gene expression. We don't see any difference in the gene expression of the symbiont across the host life stages, which suggests perhaps most of the regulation occurs through the host gene expression. We see genes associated with tyrosine production and transport in the pupal stage, and these are more highly expressed in symbiotic larvae and pupae. We see symbiont-specific sugars being produced by the host at the pupal stage, and potentially the recruitment of an insect-specific sugar, trehalose, to facilitate that growth of the symbiont. And then finally, we see genes associated with lysosomes and phagocytosis, which may be involved in symbiont reorganization and prevention of overproliferation. So I just want to say thanks to everyone for coming, and to my lab and our funders, and also the ants, because I couldn't do it without them. And yeah, I welcome any questions. Yeah, no, that's a really good question. Obviously something we're very cognizant of in this study. So one thing we did do was, when we did the heat treatment, we left them for 4 to 6 weeks after the treatment, so that it was new queens that were actually going through that we were extracting RNA from and doing the analysis with. So originally we planned to do some of this with wild-type symbiont-free ants, because you actually get that in the system. So in the wild, there are some colonies that don't have the symbiont, which is really interesting dynamic as well. But they're very hard to find, so we weren't able to use that as a control as well. Okay, thanks so much for listening, guys.
Hello, everybody. I am Ceron Romero. I'm a postdoc at Case Western Reserve University. I'm going to be talking here about genome architecture playing an important role in population diversity and clade evolution in Cynorhizobium malilori. So, Cynorhizobium malilori is a nitrogen-fixing bacteria. It basically forms these nodes in the roots of the host, Medicago truncatula, where basically it fixes the nitrogen and helps the plant to grow. This bacteria has a genome that is composed of a chromosome and 2 big replicons, which are PCMA, a megaplasmid, and also PCMB, a chromid. And the difference here is basically that a megaplasmid was first of all a plasmid, and then it basically through time it accumulated sequences to a point that it now is very big. And the chromid is sort of like the opposite dynamics, right? Probably was a chromosome that is being losing sequences through time, to a point that now is very similar in size to the other replicon, to the megaplasmid. Now, the type of evolutionary events that I'm interested, or that we were interested in this particular work, was a horizontal gene transfer, detecting horizontal gene transfer. And here are some important definitions that I'm gonna be basically applying for the rest of this talk. When you have a DNA coming into the cell, which can happen in bacteria for those mechanisms that you're seeing in the top, what you can see is that the DNA, once it's inside the cell, it can be incorporated into the genome by either homologous recombination, where basically what you have is that there is a replacement of an allele, or what you can have is a legitimate recombination, which is basically when you have that the DNA that comes from the donor to the recipient is actually representing a complete new different gene. Now, the aim of this particular work was to study the impact of HGT in both clade evolution and also the population structure of Caenorhizoium malilori. And then for that, we use a sample that was composed of 191 strains of Caenorhizoium that were sampled across the geographic range of the host. And from there, we took 18 strains that we use for long-read sequencing. Now, with the bigger data set with 191 sequences, what we did is to reconstruct the phylogeny. For that, what you're seeing there is the consensus tree of 4 different species trees that we came out with. And for that, we use basically different approaches, maximum likelihood, we use parsimony, different data sets, the core genome, concatenation of coding sequences. Anyways, the idea here is that with this phylogeny, what you can see is that there are at least 5 distinctive clades. That's in the chromosome, but when we do the same in the PCMA and the PCMB, the other 2 replicants, what we see is that there's a lot of discordance. So this by itself, it demonstrates that at least there's a lot of chances that there's a lot of homologous recombination involving these 2 replicants. Now, how we detect horizontal gene transfer? So there are many different methods. Probably the more famous or the more widely used are with phylogenetics, basically comparing gene trees against species trees, and then the discordance, as the one that you can see here in the bottom, are potential cases of horizontal gene transfer. Other methods that are normally used are detecting those regions in the genome that have a very distinct or different sequence composition in comparison with the rest of the genome. But that being said, none of these methods really work for us. And in part, this was because these strains have are very closely related. So those genomes are very, very similar. And when you have a data set like that, gene trees are very unreliable. And also, this type of mechanisms, like methods like the ones based on sequence composition, normally have very, like this, let's say, diffuse results, like very difficult to get some information from it. Now, what did work for us was, for instance, this method or tool, clonal frame ML. The purpose with this tool is to detect those substitutions that normally have an impact on the phylogeny. And it basically detects one of those substitutions are actually coming from homologous recombination with external sources. And this is actually very important. When I say external sources are strains that are not part of the sample. And it differentiates those from the substitutions that are actually coming from the mutation that is expected under vertical inheritance. Now, the other thing that did work for us was PopCogent. And with this tool, the idea is to detect those homologous recombination cases in which is homologous recombination with internal sources. So this is the difference with the other method. Here, the idea is to detect homologous recombination that happens across the strains that are composing the data set. What is very cool with this tool is that it actually detects a particular type of effect of the homologous recombination, and is when you have a sequence that is entering or that is basically transferred to a genome, sometimes this sequence is possibly selected, and then it sweeps through the population, and it causes this homogenizing effect. So the idea here with this tool is the longer that you have these fragments that are homogenized, the more evidence that you have of homologous recombination. And then if you account of homologous recombination as gene flow, then, for example, when you construct a network with this type of information, you can see that there are subgroups of these strains that share more or that do more homologous recombination among them than with the other strains. So it's sort of like give you information about the population structure in your sample. So these two are very complementary. So here I'm going to show you some of the results that we got. This is with clonal frame ML. The idea is here I'm showing you, this is the phylogenetic tree that it calculates, and it's basically totally consistent with the phylogenetic tree that we got. And here is the alignment of the core genome. So now this has a lot of colors. What I want you to focus here is in the dark blue, because those are the regions that clonal frame detected as cases of homologous recombination with external sources. And as you can see here, a lot of these potential cases of homologous recombination are actually placed in the clades 4 and 2, which are these green clades and purple clades. But at the end of the day, what we really understood from using this tool is that there's a very distinct patterns of homologous recombination across different clades. Now, using PopCogen, the other tool, what we found is that what we found here is that some level of consistency. What we found with the other tool here, once again, this green clade, there's a lot of recombination among all these strains that compose this green clade, but not much between those strains and the other clades. We didn't see that with the purple clade, right? So this basically is saying these guys are having a lot of recombination with internal source, the green clade, and the external source, but the purple clade is more about recombination with external sources and not much with internal sources. Now the graph that you're seeing in the bottom is like a summary of the network, right? The x-axis, the y-axis is showing you the amount of recombination, and then the x-axis is just showing you the different clades. And you can see there two type of box plots. The like reddish, I don't know how to describe that color, but one of them is basically the intraclade recombination, and the other one is the interclade recombination. So as you can see there, the clade 4, which is basically the green clade, has way more intraclade recombination than interclade recombination, and that's basically why PopCogen determined that this is a subpopulation. So overall, we understood from this tool that this is basically telling us our sample had a population structure, and that homologous recombination is very useful for actually detecting that population structure. Now, when you do that with the plasmids, I'm not showing now. Okay, here we go. With the plasmid, with the PCMA and PCMB, then you actually don't see like really anything very important in those summary graphs, and the networks are showing a lot of recombination everywhere, right? So basically what that's saying is, once again, if you do this and you track the homologous recombination in the chromosome, then you're gonna see population structure, but once you move to the PCMA and PCMB, the chromid and the megaplasmid, there's just recombination everywhere. Now, what we decided to do next was to see if we take all the different replicons, PCMAs, PCMBs, chromosomes, and we put them all together and calculate how much recombination we have among all those replicons, and try to see what is the direction of that recombination. This is basically what we got. So we got a ton of recombination between the PCMBs and the PCMAs, but probably what is more interesting here is that there's a ton of recombination between the chromosome and the PCMAs, but not much between the chromosome and the PCMBs. Now, we really don't know for sure why is this, why there's more recombination between the chromosome and the PCMA and not with the PCMB, but this may be related with the fact that they have just very different evolutionary histories, right? One of them, the PCMB, has more like a deletional bias. The other one, the PCMA, has more like insertional bias. But again, that's something to test. Now, what we decided to do next was, okay, let's just detect those genes that are involved in these events of recombination, and let's see what are the functions. And then for that, we use the long-read sequence data. Let me see if I can show you. Okay, for that, we use the long-read sequence data. And here the idea was just basically to track those genes that are shared among pairs of these replicons. Let's say between the genes that are shared between the PCMA and the PCMB, the genes that are shared between the chromosome and the PCMA, and so on. If we count the genes and the sequences, right, because genes or gene families have different amounts of sequences, so if you count for genes and and sequences, and you divide them by function like this, what we saw more interesting is those genes that are shared among all the replicons, the ones that are on the top. Those are basically the genes that are basically shuffling around all the replicons. And what you can see there is that most of them are actually transpossesses. And what is very interesting is that when you see the second part of the graph, which is basically the sequences, you can see that there's more of them in the PCMA and the chromosome, right? So, yeah, I mean, what we have from this presentation basically is two take-home messages. First of all, that different clades have very distinct patterns of homologous recombination, and this informs us that there's a population structure in Cenon malilori. And the second one is that there is a mechanism, or there seems to be a mechanism that is based on transpossesses that facilitates HGT, but is mostly between PCMA and the chromosome in Cenon malilori. So with that, I would like to thank the HIT Lab and all the different institutions that promoted this work, and also, of course, you for your attention. And I will take questions.
Hi everyone, my name is Franciane Cedrola, and today I will present the results of this work that I am developing. You can hear me? Yes, that I am developing in my postdoc in University of Campinas, Brazil. Genomics of ciliates from rumen and secant of herbivorous mammals, a functional comparative approach. Over the evolutionary history, several herbivorous mammals had established symbiotic associations with protease ciliates in their gastrointestinal tract. And most of them represented in these drawings, and in my opinion, the most beautiful things in the world, are mutualists and participate in several ways in the digestive metabolism and other aspects of the metabolism of their hosts. Despite the increasing knowledge about the metabolic features of these organisms in the last years, little is known about whether there are differences in the metabolic profile of ciliates associated with distinct gastrointestinal environments. Today we have two kinds of mammals that aggregate ciliates in their gastrointestinal tract, the 40 gut fermenters, which are represented mainly by some Artiodactyla species, and the hind gut fermenters, which are represented mainly by some Perissodactyla and Rodentia species. The 40 gut fermenters present the fermentative chamber, mainly the rumen located before the region of abscission in the gastrointestinal tract, which is the duodenum. And the hind gut fermenters present the fermentative chambers, mainly the colon and the cecum, located after the region of abscission in the gastrointestinal tract. And besides these differences in location, these chambers are very similar physicochemically. And they are both anaerobic environments with pH around 7 and the temperature ranging from 38 to 42 degrees Celsius. Most of the studies about the microbiome associated with rumen, with herbivorous mammals, gastrointestinal associated with the gastrointestinal tract of herbivorous mammals are focused in the prokaryotes in the prokaryotes, and the eukaryotes are still poorly understood. And concerning the rumen ciliates, most of the studies are concentrated in rumen symbionts, and little is known about hindgut fermenter symbionts and if there are differences between hindgut and foregut symbionts. Thus, this study aims to find out what is the functional profile of ciliates inhabiting rumen and cecon of herbivorous mammals. For that, we sequenced and assembled the genomes of 15 ciliates isolated from distinct gastrointestinal environments, the rumen of cattle and the cecon of a capybara. And these 15 genomes were joined to other 17 genomes available in any CBI database, totalizing 32 genomes in which we performed the gene prediction and functional annotations. The functional annotation was performed in two distinct ways, in a general way using the software GNOG, and specifically for the carbohydrate metabolism of the ciliates using the software DBCAM, which identify the carbohydrate active enzymes present in the genomes analyzed. So, concerning the general metabolism, which is the GNOG annotations, we do not observe significant differences among ciliates inhabiting rumen and cecon, as we can see in this graph, representing the proportion of cluster of orthologous genes identified in the genomes analyzed. And most of these genes are inserted in metabolic pathways related to the host digestive metabolism described here, which emphasize the mutualistic character of these ciliates analyzed here. However, although we do not observe significant discrepancies in the metabolic profiles of these ciliates inhabiting these two distinct environments, we observe distinct functional profiles when you analyze specifically the carbohydrate metabolism. In a general way, we found 4,243 enzymes in these genomes, and they are annotated across different families involved in carbohydrates breakdown and synthesis listed here. And most of these enzymes are related to the energy metabolism of the ciliates and acted in distinct substrates, like complex non-structural carbohydrates marked in these graphs in blue, complex structural carbohydrates marked in yellow, and microbial cell wall carbohydrates, mainly peptidoglycan and chitin, marked here in green. And we can see easy two distinct profiles among ciliates, between ciliates inhabiting rumen and cecon environments. When you analyze these two environments in a comparative way, you can see that rumen ciliates presented more enzymes proportionally than the cecon ones, and most of these enzymes are specialized in structural carbohydrates, and most of the enzymes annotated in rumen ciliates are enriched. The cecon ciliates, on the other hand, presented less enzymes than the rumen ones, and most of these enzymes are specialized in non-structural carbohydrates. Taking into account these environments in a more specific way, first the cecon, we could observe two functional groups within this environment, one represented by the family Cyclopodeidae, with ciliates specialized mainly only in non-structural carbohydrates, and the other represented by the family Pycnotrichidae, with ciliates specialized only in structural carbohydrates. These enzymes annotated are used in the ciliate metabolism, and the main products are short-chain fat acids, which represent the mainly energy source for herbivorous mammals. And these short-chain fatty acids are absorbed in the cecon or during the cecotrophy process. And when you analyze the metabolic profile of these ciliates in a more detailed way within families, we do not observe any metabolic diversity intra-family in ciliates inhabiting the cecon. When you analyze the rumen specifically, we also observed two functional groups, two major functional groups, one represented by the family of Roscolestidae, with ciliates specialized mainly in structural carbohydrates, and the other represented by the family Isotrichidae, with ciliates specialized mainly in non-structural carbohydrates. And as in cecon, these enzymes are used in their own ciliate metabolism, and the enzymatic activity, the products of their enzymatic activities are SAFs, which are absorbed in the rumen and in the duodenum. But when you analyze the metabolic profile of the ciliates within family in rumen ciliates, we observe the distinct functional profiles. For example, the first functional group observed in our study, the family Isotrichidae, is subdivided in two genera: the genus Dasytricha, which ciliates specialized in structural carbohydrates, and the genus Isotricha, which ciliates specialized only non-structural carbohydrates, as we can see in the graphs, distinct functional patterns within the family Isotrichidae. In the same way, when you analyze the family of Roscolestidae in a more detailed way, you observed three groups that this family is subdivided in three subfamilies: the subfamily Antogenini, which ciliates specialized only in non-structural carbohydrates, and the subfamilies Diplogenini of Roscolescini, which ciliates specialized in structural carbohydrates. As and we can see, we have a great metabolic diversity within rumen ciliates, and thus they are more diverse metabolically than their relatives inhabiting the secund environment. This metabolic diversity observed in rumen ciliates may represent is crucial for maintaining the balance of this ecosystem, because it allows niche partitioning among ciliate species, with each taxonomic group specialized in a particular kind of substrate. And in fact, ruminants in previous studies are pointed out as more diversified than other herbivorous mammals, and these evolutionary success are attributed mainly by for the rumination, which is a peculiar behavior present in these animals, which guarantee the mastication of food, the better mastication of food, and consequently a better viability of food sources for the distinctive microbial taxa associated to this environment, and this guarantee an efficient digestive process consequently. Moreover, rumen ensures more efficient digestion because it is located before the site of nutrient absorption in the gastrointestinal tract, which is the duodenum. And in this case, the digestion do not return to the digestive tract after the fermentation, like in hindgut fermenters. And these animals are not dependent of coprophagy and secotrophy, and thus they don't have food intake limitation, and they are more efficient because of that. And the rumen ciliates. The rumen ciliates, as the ruminants, they are also considered more diversified than the relatives inhabiting other inhabiting the secund and other hindgut fermentation chambers, as we can see in this graph, showing the differences in speciation rates between hindgut symbionts and rumen symbionts. And these differences in speciation rates may be related to the transition from the hindguts to the rumen during the evolutionary process history of these organisms, because the hindgut was reconstructed as the ancestral environment of these organisms. Thus, the transition to rumen may represent a key factor for the explosive irradiation of rumen ciliates, and occurred at the same time of the beginning of the diversification of ruminants. And our genomic data corroborate these previous studies, as rumen ciliates exhibit greater metabolic diversity than cecal ciliates. As some final remarks of this work, we have that endosymbiotic ciliates of herbivorous mammals exhibit significant functional and metabolic diversity between different gastrointestinal environments evaluated. Specifically, the rumen ciliates exhibit greater metabolic diversity compared to cecal ones. And finally, our findings support theories on the transition and diversification of ciliates from hindgut to rumen environment, which may have represented a key evolutionary step that facilitated the adaptive radiation and success of ruminant mammals. I would like to thank you, the Laboratório de Diversidade Genética, and my colleagues, Laboratório de Protozoologia, Instituto Estadual de Florestas, and Instituto de Zootecnia for the samples, FAPESP, CAPES, and CNPq, and you for your attention. Thank you.